Hi everyone, my name is Yousif Aljalali and today I'll be presenting on Dr. Jennifer A. Doudna, a molecular biologist and biochemist who helped revolutionize biology with the discovery of CRISPR-Cas9, a tool that allows scientists to edit DNA with precision. I chose Doudna because her story combines curiosity, perseverance, and responsibility, which are values that I really identify with as someone interested in biology and healthcare. Alright, so let's talk about her early life. Jennifer Doudna was born in 1964 in Washington, D.C., and grew up in Hilo, Hawaii. Natural environment there, rainforests, volcanoes, and ocean life helped spark her fascination with how living things work. Her interest in scientists really began in sixth grade after reading The Double Helix by James Watson, which told the story of DNA's discovery. So eventually she went to college and earned her biochemistry degree at Pomona College and then went on to Harvard Medical School for graduate studies. After that, she did postdoctoral research at the University of Colorado Boulder, where she focused on the structure and function of RNA, which was research that laid the foundation for CRISPR. So she also faced a lot of challenges as along her journey. In high school, her counselor told her that girls don't do science, which could have discouraged her, but instead it motivated her to push even harder. Later, as a woman in a mostly male field, she often felt isolated and unsure of herself. Still, she stayed persistent and relied on mentors who encouraged her to keep going. Alright, so let's talk about her hobbies. Growing up, Doudna loved reading, hiking, biking, and exploring lava flow caves in Hawaii. She was always drawn to nature and discovery. Now she still enjoys reading, hiking, and gardening, which she says helps her relax and think creatively about her research. Outside the lab, she's also passionate about mentoring youth scientists and advocating for women in STEM. What I admire most about Jennifer Doudna is her curiosity and her sense of responsibility. She's inspired by the natural world and uses that inspiration to make discoveries that directly improve people's lives. When CRISPR's power became clear, she didn't just celebrate in the discovery, she started leading ethical discussions about how it should and shouldn't be used. That balance between innovation and integrity really stands out to me. Like her, I'm drawn to science because I see it as a way to help others, and she's the perfect example of using science and knowledge for good. Let's talk about her field. Doudna is a molecular biologist and biochemist who studies gene expression and gene editing. Her lab investigates questions like how do mRNA molecules function inside cells? How can we edit genes safely and precisely? How can CRISPR be used to treat genetic diseases? And what are the ethical limits of altering DNA? Her work connects directly to core biology topics we study: DNA, RNA, proteins, and how small molecular changes can have huge effects on health. Alright, one recent paper from Doudna's lab, published in Cell in 2024, is titled Rapid DNA Unwinding Accelerates Genome Editing by Engineered CRISPR-Cas9. In this study, her team compared wild-type Cas9 to an engineered Cas9 that unwinds DNA faster. They used plasmid transfection to deliver Cas9 into cells, a fluorescent reporter assay using the EGFP gene, and flow cytometry to count edited cells. The results showed that the engineered Cas9 knocked down EGFP much more efficiently than the wild type. This means that the faster DNA unwinding makes CRISPR editing more precise and powerful, showing how small molecular tweaks can dramatically improve gene editing tools. So I have the results right here. Doudna's work has uncovered how CRISPR-Cas systems function in bacteria and how they can be adapted for use in human cells. Her discoveries have already led to potential treatments for genetic diseases and cancer. Currently, her lab focuses on improving CRISPR delivery inside living organisms, reducing off-target effects, and building ethical and regulatory frameworks for responsible use. She's continuing to lead both in science and shaping on how biotechnologies are handled worldwide. And here are my references. Thanks for watching.